Bacillus species, they are known to be large aerobic gram-positive rods in chains. Spores are located in the center of a non-motile bacilli. Culture gelatin stubs or resembles inverted fir tree. They are known to be saprophytic organisms, prevalent in soil, water, and air, and also on vegetation. Example of these are Bacillus cereus and Bacillus subtilis. Bacillus thuringiensis seen in insect pathogens. Both Bacillus cereus and thuringiensis may occasionally produce diseases in immunocompromised humans. Bacillus anthracis can be seen in animal products, usually herbivores. Bacillus cereus. It is known to cause food poisoning. It has two distinct forms. The emetic type is associated with fried rice or pasta. It is manifested by nausea, vomiting, abdominal cramps, occasionally diarrhea. It is self-limiting, with the recovery occurring within 24 hours. These manifestations begins 1 to 5 hours after ingestion. The diarrheal type has an incubation period of 1 to 24 hours and it is manifested by profuse diarrhea with abdominal pain and cramps. Fever and vomiting are usually uncommon. Bacillus anthracis causes anthrax, which is primarily a disease of herbivores. Anthrax is endemic among developing countries. Because they are non-saprophytic organisms, the spores can be found on contaminated soil vegetation. In animals, the portal of entry is the mouth and gastrointestinal tract. In humans, the infection is usually occurred by entry of spores through the injured skin, known as cutaneous anthrax. Uncommon on mucous membrane, gastrointestinal anthrax, or by inhalation of spores into the lungs, inhalation anthrax. Once the spores germinate, the bacilli spread via lymphatic to the bloodstreams, gelatinous edema and congestion form. Anthrax toxins are made of 3 proteins: protective antigen, edema factor, and lethal factor. Edema factor is an adenylate cyclase. Together with protective antigen, it forms a toxin known as edema toxin. Lethal factor plus protective antigen form a lethal toxin, which is a major virulence factor and can cause of death. Anthrax toxin genes are present on a plasmid. PXO2 is a capsule gene. Wool sorter's disease or inhalation anthrax. Inhaled spores will result to lung germination and later on can cause hemorrhagic meningitis and sepsis. In humans, approximately 95% of cases are cutaneous anthrax and 5% are inhalation. Generally occurs on exposed surfaces of the arms, hands, followed in frequency by the face or neck. A pruritic papule develops 1 to 7 days after entry of organisms or spores through a scratch. Initially, it resembles an insect bite. The papule rapidly changes into a vesicle or small rings of vesicle that coalesce, and necrotic ulcer develops. The characteristic of this lesion is a central black eschar. A marked eczema occurs, lymphadenitis, and lymphadenopathy. Systemic signs and symptoms of fever, malaise, and headache may occur. After 7 to 10 days, fully developed eschar formed. It dries, loosens, and separates. Healing is by granulation, and it leaves a scar. Inhalation anthrax, 6 weeks incubation period. The early clinical manifestations associated with marked hemorrhagic necrosis and edema of the mediastinum. Substernal pain may be prominent. Mediastinal widening on the chest X-ray can be seen. Hemorrhagic pleural effusions and cough can be noticed. Later on, it will result to sepsis, bowel ulceration, and meningitis. While on diagnostic laboratory test, a blood agar plates characterized by non-hemolytic gray to white, tenacious colonies, rough textures, ground glass appearance, and a medusa head or a curly hair. Definitive identification of Bacillus anthracis is fluorescent antibody and PCR. Many antibiotics are effective against anthrax in humans, but treatment must be started early. Ciprofloxacin is recommended for the treatment. Penicillin G along with gentamicin and streptomycin has previously been used to treat anthrax. For control measures, it includes disposal of animal carcasses by burning or depural in lime pit, decontamination using autoclaving, protective clothing gloves by handling potential infected materials, active immunization of domestic animals, and usually persons with high occupational risk should be immunized.